Good day, everyone. My name is Kailas Radhakrishnan. I am a third course medical student of Kharkiv National Medical University. Today, we will discuss about one of the most important and major genetic defect, which is called the Huntington's disease. Today, we will discuss about introduction, a small introduction about genetic diseases, about Huntington's disease, its genetics, genetic mutation, how it is inherited, its mechanism, function of Huntington protein, cellular and macroscopical changes which occurs during Huntington's disease, symptoms, diagnosis, its management, and conclusion. So let's start with a small introduction about genetic disorders. So genetic disorders occurs when a mutation affects your genes or when you have the wrong amount of genetic material. Genes are made up of deoxyribonucleic acid, which contain instructions for cell functioning and characteristics that make you unique. You receive half your genes from each biological parent and may inherit a gene mutation from one parent or both. Sometimes genes change due to issues with DNA. This can raise your risk of having a genetic disorder. Some cause symptoms at birth, while others develop over time. Genetic disorders can be chromosomal, complex or multifactorial, and single gene or monogenic. Chromosomal, this type affects the structures that hold your genes or DNA within each cell, which is called as the chromosomes. With these conditions, people are missing or having duplicated chromosome material. Um, Next one is complex or multifactorial disorders stem from a combination of gene mutations and other factors. They include chemical exposure, diet, certain medication and tobacco or alcohol use. Single genic or monogenic, this group of conditions occur from a single gene mutation. That's all about a small introduction about genetic diseases. Now, let's talk about today's topic, most important topic, Huntington's disease. So Huntington's disease, it is also known as Huntington's chorea. It is a neurodegenerative disease that is mostly inherited. It is caused due to a single defect gene in chromosome number 4. The earliest symptoms are often subtle problems with motor and mental abilities. Um, a general lack of coordination and unsteady gait often follows. It is a basal ganglial disease causing hyperkinetic movement disorder known as the chorea and inherited disease that causes the progressive breakdown of nerve cells in the brain. This disease has a wide impact on person's functional abilities and usually result in movement, thinking, and psychiatric disorders. As this disease advances, uncoordinated involuntary body movements of chorea become more apparent. The physical abilities gradually worsen until coordinated movement becomes difficult and the person is unable to talk. Mental abilities generally decline into dementia. The specific symptoms vary somewhat between people. Now, let's talk about genetics of this disease. So everyone has two copies of Huntington gene, which codes for the Huntington protein. This Huntington gene is also called the Huntington disease gene. It's a part of the gene is repeated section called trinucleotide repeat expansion. It's a short repeat which varies in length between individuals and may change length between generations. If the repeat is present in a healthy gene, a dynamic mutation may increase the repeat count and result in defective gene. When the length of this repeated section reaches a certain threshold, it produces an altered form of protein called as the mutant Huntington protein. The different function of these proteins are the cause of pathology changes, which in turn cause the disease symptoms. Now let's look at the genetic mutation. So the HTT gene, the HTT gene is located on the short arm of the chromosome 4 or at the 4p16.3. So the HTT contains sequence of 3 DNA bases, which is cytosine, adenine, guanine. It is repeated multiple times and is known as the trinucleotide repeat. CAG is the 3-letter genetic codon for the amino acid glutamine. So a series of them result in production of chain of glutamine known as the polyglutamine tract and the repeated part of the gene polyQ region. This slide demonstrates about the classification of trinucleotide repeats and resulting disease stages depending on the number of future repeats. As you can see in this table, if the repeat count is less than 27, or it ranges from 27 to 35, the disease will not be affected and the risk of offspring is zero. But if the repeat count ranges from 36 to 39, it is called as the reduced penetrance. And the disease status, no, the disease may or may not be affected, and the risk to offspring will be 50%. If the repeat count exceeds 40, it is called as the full penetrance. There is no doubt about the disease, it will be affected, and the risk to offspring will be 50%. Generally, people have fewer than 36 repeated glutamins in the polyQ region, which will result in the production of the cytoplasmic protein Huntington. However, a sequence of 36 or more glutamins result in the production of a protein with different characteristics. This altered form, called the mutant Huntington, increases the decay rate of certain types of neurons. So the trinucleotide CAG repeat numbering over 28 are unstable during replication, and this instability increases with the number of repeats present, and this usually leads to new expansions as generally pass. Instead of reproducing an extra copy of trinucleotide repeat, this causes the number of repeats to change the successive generations such that an unaffected parent with an intermediate number of repeats, is 28 to 35, already used penetrance ranges from 36 to 40, may pass on a copy of the gene with an increase in the number of repeats that produces fully penetrance this Huntington's disease. And also the earlier age of onset and greater severity of disease in successive generations due to increase the number of repeats is known as the genetic anticipation. The instability is greater in spermatogenesis than the oogenesis. Maternally inherited alleles are usually of a similar repeat length, whereas paternally inherited ones have a higher chance of increase in length. Rarely is Huntington's disease caused by a new mutation where neither parent has over 36 CAG repeats. Now let's talk about inheritance of Huntington's disease, how it is inherited. So Huntington disease has an autosomal dominant inheritance, which means the affected individual typically inherits one copy of the gene with expanded trinucleotide repeat from an affected parent. Since the penetrance of mutation is very high, those who have a mutated copy of gene will have the disease. In this type of inheritance pattern, each offspring of an affected individual have 50% risk of inheriting the mutant allele, so are affected with the disorder. This probability is sex-dependent or sex-linked genes or traits that are found in the X and Y chromosomes. In this pedigree chart, we could find, yes, this disease is inherited in autosomal dominant fashion. The probability of each offspring inherited and affected gene is 50%. Inheritance is independent of sex, and phenotype does not skip generations. In the rare situations where both parents have an expanded Huntington disease gene, the risk increases to 75%. And when either parent have two expanded copies, the risk is 100%, which means all children will be affected. Individuals with both genes affected are rare. For some time, Huntington disease was thought to be the only disease for which possession of second mutated gene did not affect symptoms and progression, but it has since been found that it can affect the phenotype and rate of progression. So generally, it is typically inherited from an affected parent who carries a mutation in the Huntington gene. However, up to 10% of cases are due to new mutations. So let's talk about the mechanism. Huntington protein. Yes, I already mentioned about Huntington protein. This is responsible for this disease. It interacts within over 100 other proteins. It appears to have multiple functions. The behavior of mutated protein is not completely understood, but it is toxic to certain cell types, particularly the brain cells. The early damage is most evident in the subcortical basal ganglia, initially in the striatum, but as the disease progresses, other areas of brain are also affected, including the regions of cerebral cortex. Early symptoms are attributable to functions of the striatum and its cortical connections, namely control over movement, mood, and higher cognitive function. DNA methylation also appears to be changed in this Huntington disease. Uh, Now, let's talk about the function of Huntington. So the Huntington is expressed in all cells, the highest concentration found in the brain and testes, and moderate amount in the liver, heart, and lungs. Its function are unclear, but it does interact with proteins involved in the transcription, cell signaling, and intracellular transporting. In animals genetically modified to exhibit Huntington disease, several functions of Huntington have been identified. In these animals, Huntington is important for embryonic development as its absence is related to embryonic death. Caspase is an important enzyme which plays a role in catalyzing apoptosis, is thought to be activated by the mutated gene through damaging the ubiquitin protein system. It also acts as anti-apoptotic agent, preventing the programmed cell death and controls the production of cell brain-derived neurotrophic factor, a protein that protects neurons and regulates their creation during neurogenesis. So this Huntington also facilitates synaptic vesicular transport and synaptic transmission and controls neuronal gene transcription. If the expression of Huntington is increased, brain cell survival is improved and the effect of mutant Huntington are reduced. Whereas when the expression of Huntington is reduced, the resulting characteristics are more as seen in the presence of mutant Huntington. Accordingly, the disease is thought not to be caused by inadequate production of Huntington, but by a toxic gain of function of the mutant Huntington in the body. Let's talk about cellular changes. What changes occurred in the cells during Huntington disease? So, the toxic action of mutant Huntington may manifest and produce Huntington disease pathology through multiple cellular changes. In its mutant form, the protein is more prone to cleavage that creates shorter fragments containing the polyglutamine expansion. These protein fragments have a propensity to undergo misfolding and aggregation, yielding fibrillar aggregates in which non-native polyglutamine beta strands from multiple proteins are bounded together by hydrogen bonds. So these aggregates share the same fundamental cross-beta amyloid architecture uh, seen in other protein deposition diseases. Over time, the aggregates accumulate to form the inclusion bodies within the cells, ultimately interfering with neuronal function. So the inclusion bodies have been found in both the cell nucleus and cytoplasm. Inclusion bodies in cells of the brain are one of the earliest pathological changes, and some experiments have found that they can be toxic for the cell, but other experiments have shown that they may form as part of body's defense mechanisms and help protect the cell. So in this figure, you can see a microscopic picture of the neuron and inclusion body, which occur as part of the disease. The yellow color is the neuron, and the orange color, you can see the inclusion bodies. Now let's talk about the macroscopical changes. Initially, damage to the brain is regionally specific, with the dorsal striatum in the subcortical basal ganglia being primarily affected, following later by cortical movement involvement in all the areas. Other areas of basal ganglia affected include the substantia nigra, cortical involvement into cortical layers 3, 5, and 6. Also evident is involvement of the hippocampus, Purkinje cells in the cerebellum, lateral tuberal nuclei of the hypothalamus, and parts of the thalamus. These areas are affected according to the structure and the types of neurons they contain, reducing in size as they lose cells. Striatal medium spiny neurons are the most vulnerable, particularly ones with projections towards the external globus pallidus, with interneurons and spiny cells projecting to the internal globus pallidus being less affected. Huntington disease also causes an abnormal increase in astrocytes and also activation of the microglia. I hope you know about microglia. They are immune cells of the brain. And the basal ganglia also play an important role in the movement and behavior control. Their functions are not fully understood, but theories propose that they are part of the cognitive executive system and the motor circuit. The basal ganglia ordinarily inhibit a large number of circuits to generate specific movements. To initiate a particular movement, the cerebral cortex sends a signal to basal ganglia uh, that causes inhibition to be released. Damage to the basal ganglia can cause the release or reinstatement of the inhibitions to be erratic and uncontrolled, which result in an awkward start to motion or motions to be unintentionally initiated, or motion to be halted before or beyond its intended completion. The accumulating damage to this area caused characteristic erratic movements um, associated with Huntington disease known as the chorea, which is dyskinesia. Because of basal ganglia's inability to inhibit movements, individuals affected by it inevitably experience reduced ability to produce speech and swallow foods and liquids, which is called as dysphagia. Now, very important section, symptoms of Huntington disease. The symptoms usually start at 30 to 50 years of age, but can begin earlier than this, like in juvenile Huntington disease, in which the symptoms may occur at the age of 10 years. Once they start, the symptoms usually get gradually worse. So the early symptoms include difficulty in concentrating, memory lapses, depression, including the low mood, lack of interest in things, feelings of hopelessness, stumbling, and clumsiness. Mood swings, such as irritability or aggressive behavior. Over time, someone with Huntington disease may develop involuntary jerking or fidgety movements of the limbs and the body, difficulty speaking clearly, like eventually they may find all the communication very difficult. Swallowing problems. They may choke on food and get lung infections like pneumonia from food going down the wrong way. Increasingly slow or rigid movements, personality changes. Sometimes they may change so they don't seem like their former self at all. Breathing problems, difficulty moving around. They may eventually lose the ability to walk or sit up by themselves. In the later stages, people with Huntington disease find daily activities increasingly difficult and will need full-time nursing care. Now, let's talk about the diagnosis, diagnostic methods for this Huntington disease. So, clinically, medical imaging such as the uh, computer tomography scan or magnetic resonance imaging scan can show atrophy of the choroid nuclei early in the disease. But these changes are not by themselves diagnostic of Huntington disease. Cerebral atrophy can be seen in the advanced stages of the disease. Functional neuroimaging techniques such as functional magnetic resonance, um, imaging positron emission tomography can show changes in brain activity before the onset of physical symptoms, but they are experimental tools and are not used clinically. So genetic testing can be used to confirm official diagnosis if no family history of Huntington disease exists. Even before the onset of symptoms, genetic testing can confirm if an individual or embryo carries an expanded copy of the trinucleotide repeat, the Huntington gene that causes the disease. Genetic counseling is available to produce advice and guidance throughout the testing procedure on the implications of a confirmed diagnosis. These implications include the impact on individual psychology, career, the family planning decisions, relatives, relationships. Despite the availability of pre-symptomatic testing, only 5% of those at risk of inheriting Huntington disease choose to do so. Because of this, Huntington disease follows an autosomal dominant pattern of inheritance. A strong motivation exists for individuals who are at risk of inheriting it to seek a diagnosis. Genetic test for Huntington disease consists of a blood test which count the numbers of CAG repeats in each of Huntington alleles. And as you can see in the slide, the cutoffs are mentioned here. At 40 or more CAG repeats, full penetrance allele exists. A positive test or positive result generally refers to this case. A positive result is not considered a diagnosis since it may be obtained decades before the symptoms begin. However, a negative test means that the individual does not carry the expanded copy of the gene and will not develop Huntington disease. The test will tell a person who originally had a 50% chance of inheriting the disease if there is genes up to 100% or is eliminated. Persons who test positive for the disease will develop Huntington disease sometime within their lifetime, provided they live long enough for the disease to appear. And 36 to 39 repeats, incomplete to reduced penetrant allele, may cause symptoms. Usually later in the adult life, the maximum risk is 50% that a person with a reduced penetrance allele will be symptomatic at age of 65 and 70% at 75. At 27 to 35 repeats, intermediate allele or large normal allele is not associated with symptomatic disease in the tested individual, but may expand upon further inheritance to give symptoms to offspring. With 26 or fewer repeats, the result is not associated with Huntington disease. Now, next diagnostic method is pre-implantation genetic diagnosis. So, embryos produced using in vitro fertilization may be genetically tested for Huntington disease using pre-implantation genetic diagnosis. And this technique, where one or two cells are extracted from a typically 4 to 8 cell embryo and then tested for genetic abnormality, can then be used to ensure embryos affected with Huntington disease genes are not implanted. So any offspring will not inherit the disease. Some forms of pre-implantation genetic diagnosis, non-disclosure of exclusion testing, allow at-risk people to have Huntington disease free offspring without revealing their own parental genotype, giving no information about whether they themselves are destined to develop Huntington disease. In exclusion testing, the embryo's DNA is compared with that of the parents and grandparents to avoid inheritance of the chromosome region containing the Huntington disease gene from the affected grandparent. In non-disclosure testing, only disease-free embryos are replaced in the while uterus, while the parental genotype and hence parental risk for Huntington disease are never disclosed. That's all I need to tell about this pre-implantation genetic testing. Now about the prenatal genetic testing. So obtaining a prenatal diagnosis for embryo or fetus in the womb is also possible using fetal genetic material acquired through chorionic villus sampling and amniocentesis can be performed if the pregnancy is further along within 14 to 18 weeks. This procedure looks at the amniotic fluid surrounding the baby for indicators of the Huntington disease mutation. These two can be paired with exclusion testing to avoid disclosure of parental genotype. Prenatal testing can be done when parents have been diagnosed with Huntington disease, when they have had genetic testing showing the expansion of Huntington gene, or when they have a 50% chance of inheriting the disease. The parents can be counseled on their options, which include termination of pregnancy and on the difficulties of a child with the identified gene. In addition, in at-risk pregnancies due to an affected male partner, non-invasive prenatal diagnosis can be performed by analyzing cell-free fetal DNA in a blood sample taken from the mother via the venipuncture between 6 and 12 weeks of pregnancy. It has no procedure-related risk of miscarriage. Now, about the life expectancy of this Huntington's disease. It's very important. Life expectancy in Huntington disease is generally about around 10 to 30 years following the onset of visible symptoms. Juvenile Huntington disease has a life expectancy rate of 10 years from onset of visible symptoms. Most life-threatening complications result from muscle coordination and, to a lesser extent, behavioral changes induced by declining cognitive function. The largest risk is pneumonia, which causes death in one third with those of Huntington disease. as the ability to synchronize movement deteriorates, difficulty clearing the lungs, and increased risk of aspirating food or drink both increase the risk of contracting pneumonia. Uh, the second greatest risk in heart disease, which causes almost a quarter of fatalities of those with Huntington disease, um, is no, it is the heart disease. And next one is suicide, which is the third greatest cause of fatalities, with 7.3% of those with Huntington disease taking their own life, and up to 27% attempting to do so. Uh, to what extent suicidal thoughts are influenced by the behavioral um, symptoms is unclear, as they signify a desire to avoid the later stages of the disease. Suicide is the greatest risk of this disease before the diagnosis is made, and the middle stages of development throughout the disease, um, and also other associated risks are also there, which includes the choking due to inability to swallow, physical injuries from falls, and also malnutrition. Now let's look at the management of Huntington's disease. Um, Treatments are available to reduce the severity of some of the Huntington disease symptoms. For many of these treatments, evidence to confirm their effectiveness in treating symptoms of Huntington disease specifically are incomplete. As the disease progresses, the ability to care for oneself declines, and carefully managed multidisciplinary caregiving becomes increasingly necessary. Although relatively few studies of exercises and therapies have shown to be helpful to rehabilitate cognitive symptoms of Huntington disease, and some evidence shows the usefulness of physical therapy, occupational therapy, and also speech therapy. Uh, the weight loss and the problems in eating due to dysphagia and other muscle discoordination are common, making nutrition management increasingly important as the disease advances. Thickening agents can be added to liquids, as thicker liquids are easier and safer to swallow. Reminding the affected person to eat slowly and to take smaller pieces of food into the mouth may also be used to prevent choking. If eating becomes too hazardous or uncomfortable, the option of using a percutaneous endoscopic gastrostomy is available. And this feeding tube permanently attached through the abdomen into the stomach reduces the risk of aspirating food and provides better nutritional management. Assessment and management by speech language pathologist with experience in Huntington disease is very much recommended. So now, time for conclusion. As I said during the first part of this webinar, Huntington disease is an inherited disorder that causes nerve cells in parts of the brain to gradually break down and die. So the disease attacks the areas of the brain, and uh, in conclusion, this disease is very distressing one, and can be defined according to the genetic basis, diagnosis, symptoms, and treatment. This debilitating disease is inherited and can be detected by testing for gene that causes Huntington's disease by diagnostic methods I explained already. And also education is also important. Education for patients, yes, educating the families of individuals, society at large, who have inherited or a risk of inheriting Huntington disease, um, have generations of experience of Huntington disease, but they may be unaware of recent breakthroughs in understanding the disease and of the availability of genetic testing. So genetic counseling benefits these individuals by updating their knowledge, seeking to dispel any unfounded belief that they may have, and helping them consider their future options and plans. The patient education program for Huntington disease has been created to help uh, educate family members, caretakers, and those diagnosed with Huntington's disease. Also covered his information concerning family planning choices, uh, care management, and other considerations. So that's all about Huntington's disease. I hope you got some knowledge about this disease. Thank you for your attention and goodbye.